Good morning, distinguished guests, ladies and gentlemen. It is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with the introductions of our special guests of honor, allow me to provide a brief overview of the initiative that brings us together today, INSTAL Africa. INSTAL Africa, the Innovation Science and Technology for Action Learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. INSTAL Africa was born from a collaboration between the Foresight Institute of Research and Translation, Rwanda, FIRAT Rwanda, and its technical partner, Drosophila UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Centre, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. Next, we will move on to narrating the profile of our invited guests.
It's an absolute honor to introduce our guest speaker, Professor Acaimo González-Reyes. Professor González-Reyes is a leading developmental biologist whose extensive career has been dedicated to exploring fundamental questions in developmental biology using Drosophila melanogaster as a model system. His research spans a broad spectrum of experimental strategies, from classical genetics to proteomics, transcriptomics, biophysics, and advanced live imaging, aimed at understanding stem cell biology, morphogenesis, and axis determination. He began his scientific journey with a PhD and postdoctoral research in Professor Ginés Morata's laboratory at the Centre for Molecular Biology in Madrid, back in 1988 through 1992, where he studied homeotic genes in embryonic patterning. His work defined the phenomenon of phenotypic suppression, later known as posterior prevalence in vertebrates, and he also demonstrated transvection in the promoter of the UBX gene, contributing foundational insights into gene regulation. From 1993 to 1998, during his postdoc with Professor Daniel St. Johnston at the University of Cambridge, he pioneered studies on oocyte determination, anterior, posterior, and dorsal ventral polarity, and epithelial patterning, using the Drosophila ovary and follicular epithelium as models. He then established his own lab at the MRC Laboratory of Molecular Biology in Cambridge, before joining the Spanish National Research Council at the Institute of Parasitology and Biomedicine in Granada. There, he connected oocyte determination to DNA repair in meiosis, molecularly characterizing 2 critical genes, and began his long-term work on germline stem cells. Since 2004, at the Centro Andaluz de Biología del Desarrollo, Professor González-Reyes has focused on stem cell behavior and their niche interactions, as well as epithelial morphogenesis. His team uses genomics, proteomics, biophysics, and even mathematical modeling to uncover how niche cells communicate with stem cells, how the extracellular matrix supports tissue homeostasis, and how integrins help prevent epithelial hyperplasia during egg formation. His career truly exemplifies a deep, enduring commitment to developmental biology through bold, multidisciplinary approaches. He's shaped how we understand gene regulation, stem cell dynamics, and tissue morphogenesis. And now we'll hear from the professor himself, Professor Acaimo González-Reyes. The floor is yours.
Can you hear me well? Can you hear me?
Yes, sir, we can.
So shall I start?
We can hear you, sir.
Shall I start then?
Yes, sir, you can start, sir.
Okay. Good morning. I can't see you, but hopefully you can see me and hear me. Before I start with this, I would like to thank Kudirat for the invitation to speak here today. It's a pleasure and it's an honor to be in your company, and I hope that we enjoy talking about science together. And thank you also for preparing the introduction. I see myself more of a humble researcher than what the introduction said. So my aim in my scientific life has been to try and understand basic logical questions. This is what I would like to try today. I'm going to present two projects that we've published. One of them very recently in the lab have to do with the biology of stem cells, and in particular with the effect that aging has on the stem cell populations. That's the title of the talk. I'm going to tell you about what we found on the behavior of stem cells while they age. Now let me start with an easy questions. Stem cells, we know we all have them. We found them in our tissues, in our organs. But I would like to start with video, with a visual answer for the questions. What are stem cells here for? What sort of use do we make of them? And we see this beautiful insect, which is just escaping the predator. And even though it seems very forward video, it really, when it comes to biology, it really requires a great deal of events. First of all, the dragonfly has to see the predator, has to compute the input, has to react to it. It has to move muscles that have been attached to the cuticle, and it applies to the frog. This bullfrog is seeing the predator, is seeing the prey. Again, it's reacted to this input, and then it's coordinated a series of movements. Now, the key question here is that all these behaviors involving over 200 cell types, at least in humans, and involving again trillions of cells. All this comes from a single cell, which is the zygote, and that's a property shared most of the living animals on Earth. And then we inherit information from our parents, mom and dad, and we generate a zygote, a single cell, which is able to produce many different cell types. and they have to proliferate in a regulated and controlled manner until they form the different tissues that conform our bodies. And this is truly remarkable. The zygote, by definition, is the most powerful stem cell in our bodies. In a more formal way, stem cells, what are they? So cells that keep form our organs, and not only this, but in adulthood, once we finish our development, they provide the force to keep our organs in condition. So there's this very nice sentence or group paragraph written by one of the sinners of nature a number of years ago. And it's a pretty good example of what is the definition of a stem cell, what they do. Of course, they are the component of most of our organs. They are helpful in when we get this loss of cells throughout the life and throughout the function of the different organs. They are also used to repair damage and disease. So they're useful not only in physiological but also in pathological conditions. Now, stem cells and stem cell populations have this wonderful skill. They renew themselves. So even centenary people have plenty of stem cells and stem cell populations. Not only this, stem cells, depending on their quality, they have the potential to give rise to one, few, many, or all types of an organism. So they are truly interesting cells from the biological point of view, and we've dedicated a number of years working with them. Now, a property of stem cells, most of them are found in these specialized microenvironments, also referred to as niches. Most of the niches, cellular niches, also have acellular niches, but as I said, most of them have niche cells that basically allow the cells to prevent differentiation, and they control their proliferation. And this niche is important because they respond to factors. They can respond to local signals, like ligand that is secreted by a cell and received by the stem cells. They respond to systemic factors. Diet could be one of those. Circadian rhythm or clocks could also be another. For today's talk, it's important to know that aging also affects the function of niches and the stem cells within. Now, our model system is the little insect. I don't need, after all this that you've been hearing about Drosophila, I don't need to introduce the system to, beyond saying that it is in the female where we find the two ovaries, which are composed of tubules called ovarioles. These ovarioles are basically there to produce new eggs, which later on will be fertilized by the male sperm. Now, each of these ovarioles, this is a picture of one, is an array of developing egg chambers. So the farther you move down the variole, the larger or more developed the egg chambers are. So here you've got a series of developing egg chambers. If we go to the last one, to the most mature ones, large cell at the back is the oocyte. And the other cells with these huge nuclei, they are polyploid cells called the NAR cells. They are accessory cells which in the end will die. And the entire volume of the egg chamber will be occupied by by the oocyte, which in the case of in this will be the female gamete cell. Now, all the chambers, so the female has the property to produce eggs throughout her entire life. And this is thanks to the fact that at the interior end of the variole, we have a structure called the germarium, which is the house the cells responsible for the generation of these egg chambers, which have two main components. One of them is somatic cells. As you can see in the drawing, C stands for follicle cells. These are cells that will make the follicular epithelium surrounds clusters of 6 cells, germline cells. They are of germline in origin. These are the cells that will enter meiosis and then generate the gamete. So the follicular epithelium plus the internal germ cell is what makes up the egg chambers. As I said, we have follicles and cells responsible for the regeneration of the follicular epithelia, but we also have the germline cells, or GSCs. There are 2, 3, 4 GS per germarium, and they are surrounded by cells somatic origin, which are the ones that conform the germline stem cell niche. So these are the terminal filament cells, the cells, and the escrow cells. And it is important that you remember that these cells are the support cells of the germline stem cells. They are the ones that interact with the germline stem cells, the GSCs, and regulate their proliferation and prevent their differentiation. They keep them as stems. So what have we done with this? So we initially would like to tell you that we've developed years ago. We developed a method to film these GS issues live. So we utilized this biological glue called CSIC, and we dissected a number of variables per experiment. We attached them to this glue, and then we could film them under a confocal microscope. And we could keep them in this supplemented medium for 16 hours, no problem. So, and that's important because, as you'll see, as I'll explain later, the cycle of a germline stem cell is around 15 hours. So that allowed us to visualize the entire cycle of a stem cell. And this is what I'm going to show you here. So this is again a scheme on the left-hand side of the slide. This is a scheme of the niche, the GSC niche. We have the somatic cells, terminal element cells, cap cells, and escrow cells. And we have, in this case, represented 2 germline stem cells. You see the green structure inside the cells, the blob, the sphere, and the elongated shape are called spectrums. And these are organelles that are present in the germ stem cells themselves. And this is important. They are an important mark because it allows us to visualize and demonstrate that when a GSC divides, the division is asymmetric because both of their cells inherit different parts of the spectrum. And what you see in the middle diagram is a presentation of the changes in the shape of the spectrum throughout the cell cycle. So as you can see, a cell finishing G2 before entering mitosis is got a round spectrosome. Then in mitosis, it goes through mitosis. Then when the total cells start cytokinesis, they form what we call a plaque. Is because in the cytokinetic ring, an accumulation of spectrosome material, which grows time. Not only the one in the ring canal, but also the apical side of the cell, until they form the bar shape. They fuse, and then cytokinesis proceeds like an hour or an hour and a half later. Cytokinesis proceeds, and then you get the severing of the two parts of the spectrosome. One of them rings in the cystoplast, which is the daughter cell of the GSC that will enter differentiation, and most of the spectrosome remains the germline stem cell. It is important for you to realize.
Prof, sir.
Hello?
Prof, sir, please, it is echoing seriously from your end. Can you just help us to maybe use one device or unmute one of your devices? It is echoing seriously. We are struggling to hear what you are saying, sir.
I only have one device.
Thank you.
Better now? Better now?
Yes, you can. It's still echoing.
Can you hear me now?
Yellow, yes, I can hear you, but it is still echoing.
But I don't know how to solve this.
Okay, I think it's better now. I think it's better. You can continue, sir.
Let's see if I can just a second. Let's see if I can try one thing. Is it better now? Have I stopped the echoing?
Just continue, Prof.
Again? Sorry?
I can still hear the echo. Just continue, sir.
Okay. Sorry about this problem with the sound. So I'm telling you that these cells, germline stem cells, divide symmetrically, and one of the daughter cells will become the cystoblast and will enter differentiation or genesis and thus differentiation, and the other cell remains as the germline stem cell, and is this cell the one that remains in contact with the cap cells and the GS and the anterior escort cells. And this movie will show a complete cycle of this of a germline stem cell division. So we are seeing here this green marker, which labels the spectrosome. When it goes into mitosis, the signal goes into the cytoplasm, but then later on it will back to the spectrosome. So this is G2. Now this is mitosis, and then back into this spectrosome marker, and you can see the plaque that is separating the two cells. Now what you can see is that there is new spectrosome material going into the pre-existing spectrosome in the middle of the cytotic ring. Now it's elongated. It is the bar phase. Now they are fusing, different shape, and then is when cytokinesis resumes, and then is severing both cell types. And we go back to round G2, and we have GSC and the cystoplast. And there are a number of events place important in this scenario. One of them is that the post-abscission mid body, which is the fraction of the membrane that contains the remains of the spindle mid body, it goes into the future GSC, not into the cystoplast. And then you can see this in this movie. So these two have several the points toward the spindle mid body, and you can see that it starts moving around the membrane until eventually goes to the apical spectrosome in the GSC and remains there. It associates there and remains until the next division there. Why am I emphasizing this? Because the mid body, post-abscission mid body, has been shown in many cell types that it is important to regulate cell proliferation. So this asymmetric division of the GC is not only asymmetric because of the behavior of the spectrosome, but also because an important signaling organelle, the mid body, the post-abscission body, goes and remains with the GSC. So that's an important aspect of GSC division. And the other one has to do with mitosis itself. So this is a scheme showing, as I was saying before, that the two daughter cells at the end of the diagram, one of them inherits most of the spectrosome, and the other one enters differentiation. But before that, what we have is that both centrosomes during mitosis start dancing in cell. They move alongside the cell, and they are anchored at both ends of the cell. One of the centrosomes associates with apical spectrosome, and this ensures that the cells divide. One of the daughter cells will remain in contact with cap cells. This is the cell that precisely will stay as a GSC. And this is something that you're going to see in this movie. So here we are looking at centrosomes in yellow, chromatin is in red, and microtubules are in green. So the centrosomes move a bit until they eventually stay at both poles of the cell, and they start creating microtubules. These are centrosomes are oriented now. They start nucleating microtubules, and the nuclear envelope of microtubules creating condensation. And now it's seen that there is green inside the nucleus. That means that the nuclear envelope has 7 pores prior to mitosis. And now it's when we get the distinction between the GSCs, one in contact with the cap cells, and the cytoplast that moves 1 cell diameter away. And this is enough to commit both daughter cells different fates. One of them will remain stem cell, the other one will end differentiation, just 1 cell diameter away. Now, another characteristic of the GSC division is that the centrioles in the GSCs and the cystoplast separate before they are duplicated. And we know this because we've utilized a specific marker for daughter cell centriole, and we see that the centrosome splits in two, like 20-30 minutes after cell division. This is what is shown here. Here we have both centrosomes in the GSC. You also see a tiny blob anterior. That is a location of the centrosome to the anterior spectrosome. Now metaphase, anaphase. You see the plaque. And now immediately 6 minutes after division, you see the splitting of the centrosomes inside the GSCs. Again, this happens before central duplication, and this sets apart this cell type from most of the cells, which upon mitosis they duplicate their centrosomes and then centrosomes separate. And the last result I would like to present to you about GSC division. We also we have the genetic means to generate tumors of stem cells, and we also analyzed the behavior of these ectopic stem cells, which don't respond to niche signals, which is reminiscent of what happens in many cancers in humans. For when you have the cancer stem cells, which are deregulated, they respond to the signals that normally control their behavior, and therefore they contribute to tumoral growth. So here we have these tumors, and we look at the division of the GSCs. And I can advance to you that these cells, you'll see that they divide symmetrically, not asymmetrically as what happens in the normal age. So here we have cell goes into mitosis, loses this is the spectrosome signal, which is took over after, and we have a plaque material which is fusing the other spectrosome material, and then we also get integration of new spectrosome material to the cells. Here we go. And so we have two daughter cells about to enter into kinesis. And this is a scene here. This is how the cells are severing the connection. And you'll see the post-substitution mid-body, which is right in the middle there, and suddenly the two daughter cells, once they are separated, they move away from each other. So even though you see these incredible masses of tumoral GSCs, they are very active changing neighbors. So again, the take-home message from this experiment is that there is a big difference in the behavior of the stem cells depending on whether they respond to the niche signals or not, as in the case of these ectopic stem cells. So from this part of the talk, I would like to conclude giving you these bullet points. First of all, that the germline stem cells are divided symmetrically, as we have seen looking at the spectrosome and the post-substitution mid-body, and even though I didn't have the time to show you, we live imaging determined that this GSC is like once every 15 hours on average. But still, the time vision was quite variable. And I think this is an inherent property of the system, that the stem cells on average behave in a way, but it's a quite distinction in times of different phases and different times of division. Second, that the spectrosome cycles through different shapes. In G2, M and 1, we have round spectrosomes, but then we have the plaque, we have the bar, fusion shapes, and this connection point. Importantly, the GSC are not the cystoblast. Is the one inherits the post-substitution mid-body's important signaling, at least in other cell types. We have demonstrated this in the ovary. Also, centrosome segregation takes place in G1 prior to centriole duplication. And finally, the tumorous GSs show symmetric behavior during division, accounting for this huge growth in the number of germline stem cells in the tumors. Okay, now I'm going to change subjects and tell you a little bit about aging. So it is well known that our tissues, our organs age, and the response of our organs to different insults, such as molecular lesions, scars, epithelial damage, blood pressure, etc., is affected with age. I do not repair my tissues as effectively as my children, for instance. So the question is we wanted to understand a little bit more of the molecular details of aging, and we started looking at what happens in our niche, the germline stem cells during aging. The first observation that is important in this context is that if you look at the number of GSCs with time, you see a significant decrease in the number of GSCs with age. There's a big drop in GSC number from 2 to 3 weeks, and this drop is maintained in 4 weeks. So you go from 2.5 GSCs on average per niche to 1.5. Which is quite a reduction in GSCs. So we decided to try and understand the reason for this drop in GSC numbers, but not focusing on the genomes themselves. We wanted to understand, or we wanted to define the context, the need had something to do with decline. So we resorted to a molecular approach utilizing a cell sorter. So we expressed a marker, a GFP marker, in the cells of interest, either the terminal filament cells and cap cells, or the escrow cells. So we generated the tissue, we dissociated it, we sorted the G cells, and then the important bit is at the end of the diagram of the flow, which is we look at gene expression. But when we did the sequencing, we did it with enough depth so we could start looking at splices in events. So we could start looking at whether exons were incorporated or not, etc. And explain in more detail in a second. So let's focus on gene expression, okay? And terminal filament cell cells. We isolated them both together here in dark green. So the first thing we saw is that we got over 1,400 genes showing changes in gene expression. Roughly 50% of them were upregulated in all cells, and 50% of them were regulated in all cells. When we looked at the genealogy of the differential expressed genes, we found a number of categories. We found cytoskeleton, cell polarity, cell adhesion, neural components, including ion channels. And the funny thing is that neither the terminal filament cells nor the caps are innervated. But still, they show change in ion channels, potassium, sodium, calcium, and in molecules involved in neural signaling. That was telling us that those cells were probably communicating or generating selling synapse with the neighboring cells. And that's the property that is well known in the cap cells and the escrow cells and the genome stem cells. We published stuff on this. Here laboratory has published stuff on this. So, and cytoskeleton cell polarity, were surprised but not completely surprised, because even though terminal filament cap cells were not known to be polarized, in other words, showing apicobasal polarity, we knew they were able to emit cell projections. So cytoskeleton was probably representing those cells. And in our model, the older you become, probably your signaling capacities are diminished. We have shown this before. So we were a bit surprised to hear about neural signaling, ion channels, and cell polarity. We were not that surprised to hear about cell fusion and cytoskeleton. Now, this is gene expression. As I was saying, we also look at alternative splice. And before I give you the results, let's introduce a little bit about what is alternative splicing. So there are a number of categories that refer to alternative splicing. One of them is differential incorporation of exons. Then we use alternative splice donor sites prime or free prime accept sites. And the fourth category, important category, is intern retention. messenger RNAs can retain introns, and that generates a number of problems. So when we look at this as these categories, X, alternative splice, alternative acceptor, or donor site for intron retention, what we found is that they all become the worse you splice out introns, and that's why we get this positive delta PSI, which is an increasing percentage of splice in all the cells. And you interpret this by saying perhaps what we see is that the older cells are less able to splice the RNAs correctly. That was one interpretation. When we look at the gene ontology cellular component terms of the differentially spliced genes, what we find again cell adhesion, cell polarity, viral signal, cytoskeleton. Very similar to what we saw with the genes that differentially expressed. And that was telling us that perhaps the genes were common to both categories. And lo and behold, what we find is when we look at the genes that are differentially expressed and the genes that are shown differentially splicing, number of those, probably nearly one-fourth of those that are alternative spliced are also differentially expressed. And that pointed to a coordination between changes in gene expression and using alternative splicing. And it's important, this is important, we'll come back to this in a couple slides. Another important point in our results, not only at terminal filament and cap cells, but also we analyzed the aging of escrow cell. And escrow cells, it is curious because escrow cells proliferate in the adult, whereas TFs and cap cells don't. Escrow cells are genetically distinct. At least 3 populations of escrow cells have been defined genetically in the niche. Escrow cells morphologically are very different and are very dynamic. They send projections, a large number of projections, compared to the capsules, which are sitting there, they don't move at all. Still, number of genes that are differentially expressed in escrow cells with H is an order of magnitude lower than in cap cells. We're talking about 120 plus genes. And if you look at whereas the number of alternative splice genes that we found or events was similar to what we found in cap cells, in T cap cells. And funny enough, here the tendency of retention is just the opposite. Young cells retain more introns than old cells. So our interpretation that the older you become, less effective you are in alternative splicing your mRNAs does not hold true at least in core cells. Now, if we look at the terms, we see cell adhesion and cytoskeleton, we don't see the neural signaling or ion channels categories. So that sets apart again escrow cell, TFs, and cap cell. And the overlap between genes which are differentially expressed in escrow cells and genes that are differentially spliced, minute. It's only 5 genes. That's much lower than what we found in TFs. So from all this, we can conclude that aging of the different niche population is very different. So we looked at common themes between them. So if you look at the differentially expressed genes, TFs and cap cells, and escrow cells, you only find 40 of those, 13 of those, which are similarly related, either up or down in cells. That's a minute proportion. If you look at genes with alternative splicing, even though you get 180 genes, which are common to both data sets, so they are spliced in both TFs and capsules and escrow cells, all 34 of them are the same events, the same splicing events. In other words, same exons, the same introns, which are retained, the same alternative 5' acceptor or donor sites. So, with all this, we could conclude that the molecular signature of the aging of the different populations of the niche is quite distinct. And then, just to finish, I'll just coming back to this coordination between differential gene expression and differential alternative splicing. So I was telling you that we got a significant overlap between the things that are the differential expressed and alternative differential alternative spliced. Of those, so what we look at the alternative splicing factors defined or identified in Drosophila, which is like 84. And of those, we find that 6 are differentially expressed, 12 are alternatively spliced, 3 are both differentially expressed and show changes in alternative splicing. And these 3 muscle blind, let's move on. And I need to give you some data. We are currently working with muscle blind, but I will give you some data on MU1. MU1. What is MU1? First of all, as I was saying, it shows with age, it shows changes in the expression of the gene and in the splicing of the gene. This MU1 was identified in the worm, in the C. elegans, this nematode. Also a model system in developmental biology. Suppressor of MEK8 ANC52 number 1 is a component of the spliceosome and it's a key component for the activation of the splicing activity. It regulates the splicing of short introns, at least in vertebrates. And also in Drosophila, it is known that it's required for the adhesion, specifically the neuromuscular junctions, in the innervation of the muscles by the nerves, because it regulates fasciculin 2 alternative splicing. So what do we find about SMU1? So this is the structure of the gene. So it's got 4 introns, tiny introns like below 70 base pairs. And then what we found is that the terminal filament and capsules, second intron was significantly retained in old cells. And the threshold as well in our significance in terms of alternative splicing changes was 50%. So a percentage of splice in equals plus 15 or minus 15, so above 15 or below 15. So that shows 18, sorry. And then when we look at the expression of SMU1 RNAi in 1 week, 3 weeks, and 4-week-old cells, we find profile. We find that 1 week and 4 week more or less similar levels, but 3 weeks is gone. It's important because I remind you that at 3 weeks we have this big drop in the number of GSCs in the niche. Now we look at escort cells as well. What we find is that there is no significant changes in alternative splicing of SMU1 in escort cells. And if you look at the expression profile of the gene in the cells at 3 or 4 weeks, we see just the opposite. We see a big increase in 3 weeks. So this is yet another example of what I mentioned before, is that the molecular signature of aging escort cells and in TFCs and capsules can be completely different. And that was again a surprise for us. What else? We validated the function of ESMO1 using either mutations that we generated in the lab or using RNA interference. And here I show you results with RNA interference. If you knock down one in TFs and cap cells, what you find is that there is a drop in the number of GSCs. And here, this is a bit, I can explain this. So this is one of the beauties that we do with Drosophila. So the top control panel is an escort cell stained, visualize the shape of the cell with a GFP marker, and then on the panel is an escort cell that is mutant now is completely mutant for ESMO1. And you can immediately see that the volume of the cell is different. And we quantify this different volume. And in the graph to the right, what you find is the blue dot represents cell volumes of controls. And the purple values are volumes of mutant cells. And you can see that the distribution of the volumes is significantly different in one genotype respect to the other. So what we can conclude from this is that ESMO1 is required in niche cells to maintain a normal population of stem cells and also a normal architecture of the cells. And then just to finish, a couple of results about FAST2. This adhesion, this neural adhesion factor, it changes with time, and you can see here in these trials. 1 week you get an amount of FAST2. 4 weeks it is increased. This expression that is also not only the expression is affected, but also the lysine. So you can see that these two isoforms, protein RA and RF, have very different profiles of inclusion of these two exons. Basically, one of them is 40% of the isoforms in young flies contain this X, whereas virtually none, or less than 10%, contain them in older flies. And also, we know that ESMO1 is required for the localization and the levels of the FAST2 isoforms. Okay, so this summary of the second part of the talk that I've been giving you. First, we've demonstrated that the aging of support cells involves changes in both gene expression and alternative splicing. That there is some coordination in those changes, and surprisingly, splicing factors such as ESMO1 and muscle blind show changes in both, and that might provide a regular mechanism as to why aging affects both gene expression and alternative splicing. I've also shown you ESMO was required in the niche cells. First, to keep a normal set of stem cells. Then for morphology and also for the regulation of cell adhesion. And the core of all our studies is that the aging of different niche cells can cause with essentially different molecular traits, and that was an interesting finding. Yeah, the last slide, but it's most important is just to acknowledge the contribution of the different people, our collaborators from Barcelona, Manu Irimia, Granada, Fedrita, and in-house, people in the lab, Patricia and Gemma, participating in the live imaging of the GSCs, and the rest participated in the aging experience. And thank you for your attention.
Thank you very much, Professor, for this wonderful presentation. I believe that we must have learned one or two things, even though from my end it's a bit very patchy. Probably it's from my end here. So the floor is open for question and answer. And thank God you really made my work very, very easy, because you already summarized all the presentation. So there's no point summarizing the summary. So thank you very much, Professor. So
Yeah,
the
yeah.
floor is open. The floor is open for questions. So, Prof, while we are waiting for others to the questions to come in, can I ask that? Yes, you've given us a very good presentation, but I can say this is the good aspect of it. Can you tell us what are the major impediments that you face in the lab while doing all this, and what are the major issues that are impeding the progress of this work translating into therapy, or clinical trials and all that? Thank you.
So you're welcome. So technically speaking, even though we have access, a single female, it takes us, I don't know, 2 minutes to dissect the ovary, so probably is, and we get access to 30 to 40 niches. So the live imaging experiments can be done reasonably easy. When you start looking to the molecular characterization of the cell, that is not that straightforward. Uh, Disgregating the tissue and isolating the cells can be troublesome, uh, and that's the point. So for instance, we can't do single cell sequencing easily.
Hmm,
It's
hmm,
expensive, and they require you have to resort to bulk RNA sequence. And of course, we are missing some aspects of the problem here. That's one aspect. The other one is that TFs and cells do not divide in the adult. So this means that we cannot make mosaic study in the adult. We have to generate the mutant cells during larval stages, which is when they divide. So that prevents us from studying the loss of function of a gene only in adult. We have to use RNA interference. That's another thing. And your last part of the question was whether we can extrapolate or export our findings to other systems more closer to our medical applications. Sure. Um, it was known before we published our work. It was known that alternative splicing was essential for the stemness of stem cells, but it was not known played a key role in support cells of the niche. So now the question is open for all those labs working on uh, hematopoietic niche, brain niches, epithelial niches as the intestinal epithelium, uh, cell niches. We start looking at splicing events in the niches and see where the age. And again, we've analyzed aging, but we could analyze other stresses, nutrition, heat, uh, damage to epithelium, you name it.
Thank you very much, Professor, for the answers. We are still waiting for further questions from the audience. Uh, Please let your question come in. Let the questions come in, please. So there's a question here, Professor.
Okay.
Thank you, Prof. for the wonderful presentation. I would like to ask why Drosophila was used for this study. Thank you, sir.
For a number of reasons. First, because we are on Drosophila. Second, because human niche is a very well studied group. And many parallels with other niches. Uh, mammalian niches, for instance, fusion. We've got a number of fusion molecular which are for the proper functioning of our niche. They are present in other niches. Curing is required here, cycling is required here, index are required in our niche. All those molecules play essential roles, for instance, in the niche, in the gut niche. Another reason is the genetic manipulation that we can perform here. So we can remove, for instance, one function from single cells. And the same structure, we have the controls. We have cells which are normal and cells which are mutant. We can analyze what is with those cells. And that allowed us, for instance, to determine that escrow cells were larger in volume, the same as capsules, which is volume than the capsules. Again, the challenge now resides in how do we extrapolate it or explore this knowledge into other systems. But we probably, with our funding, we could have done this analysis. The work that I've explained to you is more than 10 years of manipulating tissues, isolating cells, doing the sequence, generating mutants, validating the results. This is not easy.
Thank you, Prof. There's another question here. How can I intern at your lab?
That's the most difficult question to answer. There are a number of schemes to get apply. Unfortunately, we don't have the money to pay for this. We have had in the past, we have had people students from yeah, 6 months in the lab. And I applied for money to the Spanish Institutional Research Council, and we got it. So if you are interested, contact us and see whether we can find money. I know there are people there which are essentially devised for people from unlimited countries coming to China and United Give them a money.
Thank you, Prof. It's one of the challenges that are facing research in generally, in terms of funds to pass the knowledge or to build capacity, particularly for uh, um, low-income countries, like most African countries, fall under this category. Thank you. So I think there are no other questions. I can't see any.
A shy audience.
So if there is no any other question, uh, okay, there's another question here, Prof. Thank you for the wonderful presentation. Introns retention is due to aging. Is there any way to reverse this retention, or have you been able to determine pathways leading to the intron retention?
Yes, that's a point that's passed us during our studies. I showed you that um, So you see the slide. This is terminal filament capsule. Uh, you see that all cells play more in vitro. So that would go with question. That would go with your question. Um, Perhaps all cells are a little less efficient in this splicing. But if you look at DSO cells, the situation is reversed. What you find is that uh, young cells feeding more intro. So I don't think you can conclude that intro retention worsens with time. This is in our and we haven't done anything else regarding this. ESMO1, it was interesting. ESMO1 in vertebrates is with partners with SMUG, and then it creates a space for small intros to be split. If you remove either SMO1 or the partners, then the split of small intros is largely reduced. It's near the end, removing those intros. We are talking about intros below 100 base pairs. Uh, Here, in the case of, we also look at, we generated ESMO1 mutant ESCO cells, because ESCO cells is where we see most of the ESMO1 cells. Generated ESMO1 mutants, and then analyzed the generation and splicing, alternative splicing in conditions in which ESMO1 activity is reduced. And we did not see an increase in retention. So what we conclude from this is that there's a situation in vertebrates and in Drosophila with respect to SMO1 and the sizing of small intros. In fact, if you look at the distribution of intro-sized Drosophila, it's different when you get in mammals. So that's why, yeah, it's not conclusive for our results with SMO1. Whether we have managed to revert this, we have overexpressed ESMO1, for instance, in those cells, in PFs and capsules. And because if you remember, ESMO1 drops on the back, drops after 3 weeks and goes back a weeks. Now you overexpress ESMO1 high levels here, and we don't see. We see a slight phenotype that we retain some of the cells, but it's not something much, and we have not analyzed intra-rotation. So again, no, fortunately I cannot answer to your question as to whether I've managed to reverse this information.
Thank you very much, Prof. In the absence of other questions, on behalf of the organizing committee, I would say a very big thank you and our deepest appreciation and for everything that you have done, for giving us your time, to share with us from your wealth of knowledge. And I believe that the participant must have learned one or two things from what you have said. And also, I believe that when next we call upon you, you'll be very willing with all your two arms open to accept our invite and also share with us what you have been doing in the lab for the past couple of years. Thank you so much, Professor. And also to our participants, thank you very much for your involvement and participation. I believe that we must have learned one or two things which we are going to disseminate in our various research communities to touch lives and then to see how together we can progress and then make humanity better. And also, my sincere appreciation goes to the organizers, the backroom staff. Thank you very much for all that you do. Putting all these things together is not easy. Thank you very much. Before we wrap up, uh, I don't know if there are other information or closing remark. Do we have Miss Rebecca Abiodun here?
Yes, sir, I'm here.
Okay, so please go ahead with the closing remark and information.
All right, sir. Thank you so much, Prof. We really appreciate the eye-opening and the inspiring section on aging of a stem cell niche that you've disseminated to us. We are so grateful. So we are also grateful for your time and, and the knowledge you've shared with us. So we also love to have you some other times. And to everyone who have participated in this program, thank you very much for being part of the event. You are recognized and your presence made it a very special one. So I really appreciate it. So all the organizers and the teams, the behind the scenes team, we see your work. We are so grateful. Thank you for making this webinar possible. So everyone, please do have a wonderful day ahead. Thank you so much.
You're very welcome.
Thank you very much.
You're welcome.